It's wonderful to have so many friends here today and colleagues for our first, if you will, thematic CTSI retreat at Purdue. As you could see, our title, Engineering Giant Leaps in Medicine, an obvious homage to Neil Armstrong, our daring alumnus, but in turn reflective of our bold desire to transform healthcare. First and foremost, I'd like to thank everyone who's been involved in organizing this event. It's going to be a wonderful day, and I appreciate all of your efforts. The broad life science industry in Indiana continues its record growth towards almost 2,000 companies, from pharmaceutical companies such as Eli Lilly to medical device companies such as Cook. The statewide impact of this sector approaches $80 billion, with almost $10 billion in exports annually. In total, it is truly the economic engine that drives our state forward. Contributors to this sector are broad, and it includes federal agencies such as the NIH, many foundations, including the Lilly Endowment and the Regan Street Foundation, as well as a host of state-supported and clinical initiatives. At the very heart of these efforts, though, are the unique collaborations of our academic institutions that are collectively what I like to call the difference makers through talent development, idea generation, technology translation, and company support. A major integrative force for our partnerships and collaborations is the Indiana CTSI. This NIH-supported institute represents our state's premier research institutions, Indiana University, including the Indiana University School of Medicine, the University of Notre Dame, and Purdue, connecting us statewide through major healthcare providers such as IU Health, so that our efforts truly do reach all of Indiana's 92 counties. It facilitates connections to life science companies to meet both workforce and technical needs, as well as supports infrastructure to help propel our discoveries into new medical approaches. Maybe most importantly, though, and I think most of you as researchers would agree, it establishes and develops interinstitutional training and research programs, which, as you know, are difficult to do, but their complementary nature and missions sort of bring out the ideal efforts in all the things that we strive for. Overall, if I think about the CTSI, I consider it a stimulus for change that has benefits clearly that go beyond our state borders. Now, many of you know our Weldon School of Biomedical Engineering, and we're a proud programmatic component of the Indiana CTSI since its inception. Our CTSI Biomedical Engineering program has shepherded over some 250 projects, moving them down the technology development pipeline, and it's everything from design and benchtop studies through preclinical studies and transfer to our company partners, both large and small. Technology licenses have spurred company growth, provided job opportunities for our graduates, generated significant royalty streams, and maybe most importantly, truly helped patients, millions of them, worldwide. These achievements continue to fuel our expansion in biomedical engineering, faculty and students at all levels, many of whom are here today. And indeed, the excitement that we have around our mission continues to pervade all that we do. My prediction truly is that the best is yet to come as we increase the collective scale of our capabilities to improve healthcare as we currently know it. We have an exciting day ahead of us. The principal goal is to connect each of you with potential collaborators, both in emerging technical areas as well as around clinical needs, in order to spur creative ideas and projects of all kinds. I encourage you to spend the entire day, if possible, learning from presentations in the morning, engaging and mentoring our young investigators during the poster session over lunchtime, and actively participating in the breakout groups this afternoon, which will be spread out all across Jiske Hall. So without further ado, it's truly my pleasure to introduce to you Theresa Mayer, our Executive Vice President for Research and Partnerships at Purdue. Professor Mayer joined us here at Purdue in August and was just appointed to the U.S. President's Council of Advisors on Science and Technology. Congratulations. She is well recognized for her research in electronic and photonic devices, as well as the successful administration of both large and interdisciplinary translational programs. We are most delighted to have her leading our research efforts at Purdue. Please join me in welcoming Theresa Mayer.
Thank you, George, and it is a real pleasure and honor to be here to kick off this event with the rest of all of you. It's just thrilling to see how many participants we have. I wasn't expecting such a large crowd, so I think it really goes to show just how many opportunities we have within the state of Indiana in this very important field of healthcare and at the intersection of engineering. So just to provide a little bit of perspective, when I came back to Purdue in August to start in this role, and I was looking at the portfolio in a deeper way, just to understand all of the capabilities and really to reach out and understand our broader partnerships with the premier research institutions, IU and Notre Dame within the state. One thing that was just extremely exciting for me to see is that within the state of Indiana, we have the CTSI, and beyond that, we have the Basic Science Cancer Center here at Purdue and the Comprehensive Cancer Center at IU. And that is a real testament to the strength of our programs, and I think, again, even more importantly, the strength of the collaboration between the institutions and the ongoing commitment. So I really would like to thank Anantha for his leadership role in being successful back in 2008 to kick off the CTSI award, and George for stepping in a leadership capacity here at Purdue, and the many others who are making this, I think, really one of, or if not the most successful CTSI in the country. And having been in two different states and different institutions, and understanding how competitive this community is and how hard other institutions are working to succeed in having such interdisciplinary programs. Again, it's just really remarkable that we're in this position, and I hope that all of you are able to use this retreat as an excellent opportunity to come together and to find the partnerships. And the CTSI is such a special award in that it really does provide that support to forge those collaborations and provide support for us to take the basic discoveries and really focus on the translation. So I hope that you all have a wonderful day here at Purdue. I guess maybe I should give a few more words just to put it into context into our greater research enterprise. So Purdue does have a very robust overall enterprise. Last year, our total research expenditures were $670 million. That has been on a progressive increase over the past 5 years. Purdue has, over the last 5 years, through the strategic initiatives, the Purdue Moves initiatives, made a major investment in life sciences more broadly, including the launch of 3 new institutes that are affiliated with this activity. Overall, I would say close to a $300 million investment, very significant investments in capital infrastructure, facilities, new faculty, and as a result of those strategic investments, we have over the same period of time seen more than a doubling in the funding from NIH. And again, the CTSI is a critical part of that. And establishing these relationships, we do hope that that continues to expand the range of different opportunities, not just within NIH, but others. George pointed to the very robust industry network, and we are not only seeing translation to our existing companies, but I think a very vibrant startup ecosystem. And again, it's very exciting to step into a role where we are having such tremendous success. Just recently, Purdue was ranked number 3 behind MIT and Stanford and within the nation for the number of startups formed. And I think, again, if you look at the numbers of startups that are being launched out of Purdue, we are seeing that a very significant number of those are in the life sciences areas and in the areas that are covered by CTSI. And so you all are not only, again, the creators of basic discoveries, but we are very active and we're supporting and enhancing the culture so that our discoveries don't just end with a publication, but we're investing in the support to see those translated so that they are of benefit to society within the state, within the nation, and the greater world. So again, I welcome you, and I hope that you have a wonderful day here at the retreat, and look forward to seeing many very positive outcomes and new collaborations that are formed.
Okay, thank you, Theresa. There's no way I'm going to figure out these microphones, but I will try. I'm now delighted to introduce to you Mark Lundstrom, our acting Dean of Engineering, while Dean Meng Chiang serves as the Director of the Office of Science and Technology at the U.S. State Department. Many of you know Professor Lundstrom as an outstanding teacher and a renowned scholar in the physics of electronic devices, as well as maybe the most sought-after collaborator and mentor on campus. He was the founding director of the NSF-supported Network for Computational Nanotechnology, so-called NCN at Purdue, and is a member of the National Academy of Engineering. Please join me in welcoming a great friend and colleague, Mark Lundstrom.
Thank you. Let's see, is my mic? This mic is working. I think we're fine. All right. All right. Thank you, George, for that generous introduction. I hope this is being recorded. I'd like to send that to my mother. So on behalf of the College of Engineering, I'd like to welcome you to this exciting day. These are exciting times at Purdue, and they're exciting times in engineering. We're one of the largest programs in the nation. In a few years, we'll be nearly 500 faculty, 10,000 undergraduates, 500 graduate students, 10,000 online students. But we aim to be much more than a large, good, affordable program. We aim to be an example of excellence at scale. We want to show how a large undergraduate program can drive excellence at the graduate level and how graduate research can enrich the undergraduate experience. So that's what we're trying to accomplish. This is also our 120th anniversary of the College of Engineering at Purdue. At Purdue, we talk about taking small steps that lead to giant leaps. We're going to be spending the year looking back and thinking about the small steps that brought us to where we are today and thinking about the giant leaps in the future. So the theme of this meeting, Engineering Giant Leaps in Medicine, is exactly the kind of thing that we're spending the year thinking about. And it's nice to kick off the year with this meeting. Now, I've been acting dean for a short time, but I've been a faculty member here for a long time. I remember when we hired George, right? And I remember when the biomedical program was a few faculty in a few departments, and it's been gratifying to see it grow into the Weldon School of Biomedical Engineering. I remember over the years seeing the IU-Purdue partnership growing, and we're really appreciative of Dean Jay Hess at the IU School of Medicine and Meng Chang, who have stepped that partnership up to a new level. So now we are really looking to increase the number of faculty and students that are involved in research, biomedical related research. There is a new clinical immersion fellowship program, a new MD, MS degree program, and a new floor to this wing that are all part of that strategy of increasing the activity in biomedical engineering. So I look forward to our keynote address, and I look forward to thinking with you about how we can engineer giant leaps in medicine. So thank you, George. Thank you all.
Thank you, Mark. And now, without further ado, it is truly my honor and privilege to introduce to you our plenary speaker, Bruce Tromberg, the director of the NIH National Institute of Biomedical Imaging and Bioengineering. We were most delighted, as I mentioned to Dr. Tromberg, when he accepted our invitation to visit us and share his vision of our field in the context of the absolutely pivotal role that the NIBIB plays as a force for innovation in what we all know as an increasingly interdisciplinary and complex field. Professor Tromberg joined the NIH about a year and a half ago, leveraging an illustrious research career in biophotonics and biomedical optics that includes the directorship of the Beckman Laser Institute and Medical Clinic, as well as the Laser Microbeam and Medical Program at the University of California, Irvine. Please join me in giving a very warm welcome to Bruce Tromberg.
Thank you, George. Thanks. Okay, it's working. We resolved. It's George's brilliance in acoustics that managed to do this for us. So thank you very much, George. Thank you for the introduction. It's really delightful to be here. I'm so happy. It's a beautiful room, and I'm impressed with all the light. I was expecting very dark and cloudy Midwestern weather. I'd like to just now spend a little bit of time echoing what you just heard from leadership here, and hopefully I'll be able to reinforce with some quantitative data some of these themes that all of you are thinking about. The first thing I'd like to do is acknowledge our team. Since I've been in the government, I've found it's extremely difficult to get people together. We don't have a budget for coffee or pizza. So, that's the big driver to bring folks together and have group meetings and so forth. So this is about half of the team. There are roughly 250 people in the NIBIB, both our intramural and our extramural program. Since I've arrived, we have 6 intramural principal investigators, and we've already expanded that with 2 new young faculty, and we'll hopefully be expanding that as well. Because, when you hear about coronavirus, for example, you hear about Ebola, immediately think of Tony Fauci and the National Institute for Allergy and Infectious Disease, and everybody sort of goes there and talks to them about what they're doing. And we need to continue to establish a presence of bioengineering on the campus at the NIH so that people can connect both with the extramural community as well as the intramural community and expand the visibility and impact of what we're doing. So hopefully this picture will grow over time. We have a senior leadership group that I'm very grateful for, and during the period of time after Rod Pettigrew left, our founding director, Jill Heemskirk was the interim director and did a wonderful job, and I've been fortunate to be able to work with and learn from her over the past year. It's actually only been a year. It seems a little longer, and you'll see when you hear all my wonky statistics why it seems like it's been a while. So as George mentioned, my background's in optics and photonics, and I came to the Beckman Laser Institute and Medical Clinic in 1988. So it opened in 1986. I was really fortunate to be in the right place at the right time. That field, I think as many of you know, has expanded tremendously, both in terms of the technologies and their translation. And I was fortunate to be able to lead an NIBIB-funded P41 center for over 20 years. That's a National Biomedical Technology Center, where we took ideas and concepts from the blackboard to the benchtop to the bedside. And we know it can be done. We had a vibrant commercialization activity. One of the really interesting advantages that we had in the Beckman Institute was a clinic in our building with an operating room and the ability to design, build, test, and deploy right in that clinic. And even if you weren't working on clinical translation, everyone knew about it. And the ambition to try to move ideas and concepts from the blackboard to the clinic, it was just sort of percolating in the airway, and it was really relatively low barrier to access that. So I think this, in some ways, prepared me for working in the government, but if you've ever worked in the government, you know you can never really be fully prepared for working in the government. It's a different type of thing than academics, for sure, and it's much more structured, so and with more of a kind of a rigid hierarchy. And I think many of the academics who come in are in shock for about a year to 2 years before they figure this out. But we have enormous power and capacity to influence things on a larger scale. And since I've grown up with the explosion of biomedical engineering and all of the associated activities with translation, I'm just very excited to be able to represent all of our community of engineers and physical scientists who are doing this and really engineering the future of health, transforming health. So I need to go back to the early days because George, I know, remembers this, and this was a big problem for our community. We like to develop technologies. We don't always have a driving biological hypothesis behind them. In fact, I would say it's rare that we have a driving biological hypothesis. And there was no place at the NIH that was willing to really support that kind of research. So our community was very active with leadership from people like Xu Chen, and others all around the country in trying to argue for the creation of a new institute. And the NIH director at the time, Harold Varmus, was actually completely dead set against it. And when the head of the NIH is absolutely opposed to it, things don't look very good. Now I'll fast forward. At late in the year, December 29th, 2000, before President Clinton ended his term, he signed a law that created the NIBIB. Now, I don't know, there may be other mysterious reasons for the creation of this law at the very end of his term, despite lots of opposition to it. I do point out that he pardoned maybe 140 and 150 people, I think on that same night, December 29th, 2000. But, I think I'd like to read the language from this law. So this is an act that was signed into law for the establishment of the NIBIB, because this is kind of our creation story. This is, I guess, like our Genesis and the first few verses from that. So I'll just, if you bear with me, the Congress makes the following findings. Basic research in imaging, bioengineering, computer science, informatics, and related fields is critical to improving healthcare, but is fundamentally different from the research in molecular biology on which the current national research institutes at the NIH are based. To ensure the development of new techniques and technologies for the 21st century, these disciplines therefore require an identity and research home at the NIH that is independent of the existing institute structure. So those words and all of the following words and that act of the creation of the NIBIB, despite lots of protests against it at the very highest levels, have allowed us to be able to do what we do. Go to the NIH with ideas that are not steeped in a biologic hypothesis and develop and engineer new technologies that are critically impactful on the future of health. So there's Rod Pettigrew with President Clinton, not at that time, but it just happened to be a shot. And Rod, I think as many of you know, he's the founding director of NIBIB, stood up the institute, and he is now the dean of a new school of medicine at Texas A&M that is engineering based. And they already have their first class. And this is the second school of medicine. So I think many of you know University of Illinois Urbana-Champaign pioneered this concept where built into the curriculum in the School of Medicine is engineering. It's a fundamental component of the academic curriculum, and it's really going to change. I think that there will be many more examples of these, particularly as they standardize the curriculum and disseminate it to other places. So what is the impact of that? Now, if we go back to before the NIBIB was created and look at the amount of money that the NIH sent to schools of engineering, this is only schools of engineering, we see 0.6% of the NIH budget, only about $100 million in the year 2000 was going to schools of engineering. Not a very good market share. So I know this isn't super impressive, but if you look at the growth rate, it is kind of impressive. Now there's nearly $800 million of the entire NIH budget. That's about 2% of the NIH budget. So that's more than a 7-fold growth that goes to schools of engineering all around the country. That's a very important change. The other really interesting statistic I'd like to point out with this is that about 30% of the NIBIB budget, about $100 million, goes to schools of engineering. Now this doesn't count physical sciences, it doesn't count radiological sciences, imaging sciences, so we do send almost all of our money to those constituencies. But very interesting that we are sending all this money. We're really the only institute where there is this is our constituency. This is the community that we're trying to grow and are growing, but we need to grow it even more and faster. Here's a very interesting statistic that I think gives you some insight into the idea that it's not just schools of engineering that are leading the charge in bioengineering. If you look at the entire NIH budget, nearly $40 billion, about 13% of it is spent on bioengineering. So obviously, more than just schools of engineering are doing bioengineering. In fact, if you look at the National Cancer Institute's budget alone, it's over $700 million in bioengineering, about 12% of their total budget. So they're right about on average for the entire NIH. And this is growing spectacularly. So in the time that this statistic has been kept over the past 10, 11 years, a 78% increase in bioengineering all across the NIH. So here's my modest prediction. I like modeling and computation, and it's really easy to extrapolate with this data. In 30 years, more than 90% of the NIH budget will be bioengineering, and I'm counting all of you to make that true. So, but you know, for those of you who've had research careers for 30 years, go back and think about the types of technologies that you used on a regular basis and how impactful they were on your life. And now think about, juxtapose that or cross-correlate that with what we're doing today. This is inevitable. And I think even my colleagues from cardiology and internal medicine and so forth, they do recognize and acknowledge. But I think the big difference is they're super happy with their technologies as they are. We are all unhappy. We're never happy with our technologies. We always want them to be better. And I think that's where we have to do a better job of communicating. Don't just say, okay, this works great. We have to tell people, imagine what we can do that will make things better, that will improve things. Every technology that any clinician is interacting with today is by definition about 40 years old. So we have new things. We have to make sure that those are developed and they get out and deployed and validated into practice. So when I came to the NIH, I was looking for the block diagram that explained everything. And here it is. There are 27 institutes and centers. It's about a $40 billion organization. We actually spend about $1.2 billion collectively in commercialization. So you can think of the NIH as a huge venture investment opportunity. And increasingly over the years, and now just over the past couple of years, we have an office that is centrally organizing all of our commercialization activities with communication. So if you submit your SBIR, STTR proposal to the wrong place, most people can identify it. So it's very important to talk to program directors so that it does get funded and it does move to the right place. Every institute has a mandate to spend a fraction of their budget on commercialization. And so this is a way to sort of help underscore that it's important to connect and discuss with program folks There is increasingly a way of thinking that commercialization is extremely important, and I'll come back to that later. But this was the original picture, and I've just modified this a little bit in the spirit of what we're trying to do. Obviously, NIBIB should be, and our technology, and this is representative of our community, should be in the middle of all this. This is the message that we're trying to communicate, that our existing technologies, while they're great, we can improve, we can always do better. So we have about $400 million in our budget and support about 1,000 grants per year, but we're heavily leveraged. We have many, many partnerships all across the NIH where we're trying to impact development in all of these institutes. Our mission is really expanding quite dramatically. I think all of you know this, you're feeling it, you're doing it, you're leading it. But just to underscore this, as I showed that statistic in the year 2000, only $100 million of NIH funds was going to schools of engineering. And for those of you who are in leadership, you know there's been a pivot. Schools of engineering have woken up and they've said, wow, human health and biology, these are hard problems. And there is incredible attention with the growth of biomedical engineering departments, but it's not just those departments. All departments are engaged. It's all across every department within schools of engineering that human health is recognized as a major challenge. Within biomedical engineering, we're adding about 1,600 new undergraduate students every year all across the country, 60 to 70 faculty per year. If you go back to when NIBIB started, we only had about 30 ABET biomedical engineering programs. And now there are, I think, 119 in 2019. There are about 130 departments nationally. So there's been this growing demand, but of course the supply and the recognition within the NIH is historically quite small. We're young. We're only 17 years old as an institute. And then, as I mentioned, the medicine engineering partnerships that we have at Urbana-Champaign and Texas A&M. And we're also doing a lot, as George pointed out, with the startups to drive not only human health, but economic growth and health. And not just in big markets, but in markets that make sense for the region or the community that you're serving. And I think this is a big, big story that I'd like to partner with you guys to help communicate and tell, because not everybody is really fully aware of this. Now, this is a very, very interesting study by Mike Kalukowicz and Dick Amen from the Mayo Clinic, where they looked at, on the y-axis, we have patents per $100 million of funding, the x-axis citations per patent, and plotted all the NIH institutes, in fact, all the federal agencies, and the size of the bubble is proportional to the size of the budget. And then they mapped this out on invention quadrants, Edison's, Pasteur's, and Bohr's quadrant. So the important thing to look at here, of course, is the position of NIBIB at the top, and actually pretty far separated from all these other organizations. So the good news is that $1 of NIH funding, which every Vice Chancellor for Research knows, is really, really good for downstream R&D investment. So this is a pretty well understood statistic, about $6 of downstream R&D investment for $1 of NIH funding. But if you pull NIBIB out, we're kind of special. And you might guess this, of course, 5-fold increased leveraging for an NIBIB dollar, because a lot of what we do is pretty upstream, it has to be de-risked, and there's a lot of work that goes on. It takes time to figure out the true applications or the true solutions of all those technologies. So this is a very interesting measurement, and these guys have argued this as a way of trying to convince Congress to divert some of their appropriations to more of the institutes that generate intellectual property and more significant downstream investment. We haven't seen it yet, but we're still hoping. So, of course, I did a local analysis of Purdue, and here are your stars here. You guys will recognize all of them up on the chart. And I've made this modest prediction. So the little over $1 million a year that's coming in should result in about $33 million, and I hope all of you can do this because I've got to be able to back these statistics up, so I'll be checking. Those should be your downstream investment dollars. But you know, we've got both Purdue PIs and subcontracts to Purdue, very vibrant activity, and you should be very proud of the work that's going on, and we'd love to see this continue to expand. We have an impact that goes beyond just the appropriated dollars that come to the NIBIB. Just like all of you, our program directors are all engineers and physical scientists, and they're all very engaged in collaborations, multidisciplinary collaborations all across the NIH. We use that as ways of generating new programs and revenue sources for our constituency. So many of you have heard of the BRAIN Initiative. We're actively involved in this. The HEAL Initiative, the SPARK Initiative, a new one that we've been leading, Data Science and Innovation in Africa, Point of Care Technologies Research Networks, supplement funds for Alzheimer's disease. So we add about $50 million per year to our budget to support research from multidisciplinary types of collaborations. Some of these are common funds, and I know that the NIH structure can be very confusing. I probably will only add to that confusion with my talk, but I'm happy to help decode some of that after my talk, and I'll be available. But more importantly, all of these program folks that we have are available and important to communicate and connect with in order to navigate through all of the complexity of the NIH. So I'd like to just point out a few highlights of all of these interdisciplinary types of collaborations. Most of you are familiar with the 21st Century Cures Act and the BRAIN Initiative. So this is a pretty spectacular statistic. $5.2 billion is projected to be the total allocation for the BRAIN Initiative. And look at this line on the bar chart in FY 2023. We're going to see I don't know, I'll believe it when I see it, a near tripling, certainly a doubling of the appropriated dollars, the base dollars of $300 million. So this is the trajectory. Only $1.3 billion has been spent so far, but here is where I'm really excited. 67% of that money has gone to technology development, to new bioengineering technologies. So I hope that to some extent inspires those of you who, if you're not doing neurotechnologies, you might want to take a look at them. They can be applied in a very beautiful way to the challenges that the BRAIN Initiative is articulating. There's another really important program in neurotechnologies called SPARK. So this is stimulating peripheral activity to relieve conditions. I would say the biggest challenge for me in the government is learning all these acronyms, and I still have a lot of cheat sheets. Some of them are exceptionally clever. In fact, BRAIN itself is an acronym, but I think it violates the principles, the laws of acronyms, because it uses BRAIN in the acronym. But anyway, I'm digressing a little bit. But SPARK is a pretty ambitious, it's a kind of Star Trek-like initiative going where very, very few researchers had gone before, designed to develop high-resolution anatomic tracing and maps of all the peripheral neural networks. It's focusing on just these organ systems, heart, lung, stomach, colon, and urinary systems. So bladder and urinary tract. It's also designed to stimulate tools for selective manipulation and observation, to translate to the clinic, and to generate data hubs, which are on the SPARK website. These are on the portal. They're actually available for, these data sets are all now up, they're shared. Every SPARK investigator has their data on that website for data sharing, manipulation, visualization, and a bunch of cloud-based tools for that. So I really encourage you to go there. And you have a SPARK expert here, Terry Powley, who has a big $10 million wonderful grant. And it's very exciting. His co-PI, Pedro, is right here. Yeah, okay. And so this is a very important program that is going and taking, I think will lead to some very beautiful and innovative but therapeutic strategies that are non-pharmacologically based that will be quite important for patients. This program is what we call a common fund program, and it's currently under discussion for whether or not it will be renewed for another 5 years. So we don't know that yet, but it is certainly ongoing in this year. And Pedro can comment on that. He probably knows more than I do. But NIBIB manages these two aspects of SPARK, the data and the tools. And I think that that's quite reflective of where all of you should be thinking about developing your approaches and your training. So here's a kind of a snapshot of the tools portfolio. 18 different tools have been developed and supported, and they're quite expansive. So what are the tools of what is the identity of NIBIB? This sort of leads me in a way to this critical point of saying, well, what is bioengineering? We hear that a lot. And what is the NIBIB? What is kind of the technical basis of your institute? So I try to sort of reduce this to roughly really 5 major conceptual groups. I'll call it 5, but the 5th one, modeling and computation, is just so embedded. It's really such a fundamental component of the fabric of everything. It may not be a separate one altogether. This is really our philosophic foundation. What makes NIBIB different from any other institute is our investigators, woven into our philosophy, is the idea that there is some kind of a symbolic representation of biologic processes or the transport of energy through biologic systems. We can mathematically represent that in a way, hopefully with analytical expressions, like ODEs and PDEs, and then we can use that to enhance how we design a biologic system or engineer biology, or how we build sensors or devices that are important for monitoring and imaging or delivering therapy. So really, at the core of what we do is modeling, computation, multi-scale approaches, machine intelligence, of course. There's been a lot of discussion about artificial intelligence. Currently, the NIH, I think, just discovered mathematics, but that's been probably the most dominant conversation that I've had over the last year and a few months. But it's clearly, obviously, embedded as such a critical component of what we do in our field. So I want to draw attention to that. We are the community that uses mathematical techniques in order to do everything else that we do. What are those other things? Well, engineered biology, and this can span from synthetic biology, which you can think of as looking at single cells as circuit opportunities, much like you can look at integrated circuit design. You can look at cells. They have switches too, like transcription factors. Those can be modeled in similar ways to electric circuits or electronic devices and reprogrammed. And so this is a very vibrant area of activity, but it also goes from the single cell synthetic biology and the reprogramming opportunities all the way to multicellular systems and organoids, microphysiologic systems, organs on chips, and so forth. We do sensors in point of care, and those span from biophotonic, bioelectronic, bioacoustic, all across the different energy forms. And point of care devices rely increasingly heavily on amplification techniques, new strategies for that, separations, microfluidics, of course. These are critical components, and they're transforming how measurements are made at the bedside with rapid response. And if you think of everybody is tracking coronavirus right now, how do we know this is coronavirus in all those patients? Every one of those patients is getting PCR, and increasingly there is this drive, this need to have on-chip identification of viruses rapidly at the bedside to be deployed all over the world. There are many, many examples of this. So sensors, point of care. We do imaging technologies. So from the biggest imaging, the biggest MRIs, all the way to, and I have an example here that Mike Garwood is developing at Minnesota, which is a head-only MRI facilitated by advances in detector technologies. We're seeing femtotesla sensitive detectors, which are facilitating entirely new MR designs as well as MEG designs. So this is obviously an area that we need to continue to advance. And then therapeutic devices, and Spark plays a big role in this, but the application of energy non-invasively. I think a really wonderful example is using acoustic wave focusing that's used in neurosurgery. So these techniques are emerging rapidly. It now has FDA approval for treating essential tremor. You can launch acoustic waves through the skull, deep into the brain, and inactivate or ablate sub-millimeter dimension locations, and even alter the blood-brain barrier so that you can change the delivery of molecules or reagents to specific sites within the brain. This is neurosurgery without scalpels. Who does not want to have that? So this is coming from our community, and it's a beautiful convergence of technologies where there's modeling, computation, imaging going on, and real-time feedback and control of those wavefronts in order to be able to achieve those important therapeutic effects. So how is all of this, that portfolio that our community is working on, and that's a kind of looking both at the community, description of our technologies, as well as looking forward and projecting to where we feel things will be important in their impact. How is that going to impact healthcare? So I like to think of healthcare right now, it's one of its limitations is it's a series of static snapshots. You take a picture with large time intervals of a variety of different things. It could be blood pressure. Most people are not looking at their blood pressure too often, and typically when they look at it, they've just driven in heavy traffic or had too much espresso, and it's hard to really know, is that the blood pressure that is the one I need to worry about with respect to my medication or my strategy for management? Certainly imaging devices are typically interacted with over large time intervals, blood chemistries, and of course looking at genomics and epigenetics and proteomics. Those things are done at very large time intervals, but we know that gene expression is dynamic and things are altering as a function of time due to epigenetic changes. So how do we integrate all of this? This is what's hoped for in the future of medicine, integrating all this information. And what's happened is that there's emerged kind of this notion that we can look at medicine on a more continuous basis, and this has helped stimulate the growth of digital health. But digital health has been more of a solution that's oriented towards consumers. I think many of you may be wearing digital health devices like Apple Watches and Fitbits and so forth that are monitoring dynamics. We know the dynamics are critically important, and we know that it's absolutely essential to bring in complex multi-parametric data because the timescale of biology is continuous. But we're just really seeing the beginning of the integration of those sensing approaches. We see motion sensing and heart rate sensing, but they're not good enough. They're good, they tell us a lot of information, but we need to do better. So the opportunity to integrate continuous sensing and dynamics with what we already have in the sensing and imaging community is really a lot of where we see the future opportunities. So if you look at the sensing and imaging technology growth, right now, if you just look at optics and photonics and radiologic systems, this is about 25% of the $450 billion per year medical device market. And much of this has grown over the course of the last 30 years. This is a snapshot. When I started in optics and photonics, the market was essentially invisible. It was in the hundreds of millions of dollars. It's now $70 billion per year. It's so ubiquitous that many of my colleagues in radiology, they often say to me, well, when is optics and photonics really going to get into medicine? So lasers that are therapeutic and a variety of different diagnostic techniques, those are all commercially available and are a $70 billion a year market. And then we've got about $30 billion or so that's been invested through venture in digital health since 2011. This has stimulated a lot of innovation, hoping that people will take advantage of this kind of information content from a consumer point of view. But there is increasingly a kind of a thin line between what is consumer and what is medical, and this thin line will vaporize in some period of time as the sensors and technologies get better and more sophisticated. And then we have an explosion of knowledge in biology. And while, of course, we're here and we talk and we love about we love the technology and we love to advance it, it's the biology where the great mysteries are. So I encourage everyone to learn as much as they can about the biology and the medicine. That's where the challenges that we need to understand to help direct our technology will emerge from. And it's not a stationary target, it's a moving target. So the advances here are just mind-boggling. They're so fast. But we need to convert that knowledge in biology to bioengineering. Technologies, devices, computational models that can help us marshal that knowledge generation in biology and then turn it into things that will be useful for human health. So we're going through right now a strategic planning exercise at the NIBIB. And I've brought in a very broad group, our National Advisory Council, other folks who are leaders throughout the country. So we met just in January, and you can see in this is our first council videocast. So if you're interested, you can check out the videocast. NIH videocasts virtually all their meetings, and you can find all the NIH meetings videocast. So you can see readouts of all these different working groups, and you sort of might imagine, based on the descriptions that I've given so far, our subgroups are data science and computation, engineered biology, sensing health and disease, imaging health and disease, advanced therapies and cures, how we move the technology development pipeline ahead, and how do we train our workforce. So we have experts from each of these areas that are involved. This is really just the beginning. The point of this strategic plan is to engage our community broadly. If you have comments on this, I'm excited to hear about them. But you'll have plenty of opportunity to comment, because as we get documents together, we'll distribute them, we'll send them out to our community, our constituency for comment, so that we can integrate that, and then ultimately have like a one-page thing I can leave off whenever I go visit people in Congress. So that's really kind of how this exercise goes. But it will give all of our colleagues and all of our community a set of common ways to define and express both the beautiful things that we're doing and their impact and future impact on human health. So we're hoping that will be the outcome of the strategic plan. So I'd like to now just kind of go through a few opportunities that are unique to the NIBIB that should be of relevance to you, that we're hoping we'll see applications from you in. I was fortunate to be able to be part of a committee when I was on the NIBIB Council that led the formation of this new mechanism called the Trailblazer mechanism. This is an R21 mechanism. It's unique in NIH because it goes for 3 years and you get a little bit more money. And what we were able to do with this mechanism is dramatically increase the number of early stage investigators. We found that R21s, because they're relatively easy to do, they're only 6 pages. So a lot of senior investigators were shutting out early stage and new investigators because they were very savvy and knew how to write 6-page proposals well. So we restructured this and said, look, it's got to have no preliminary data at all. And we create this very special Trailblazer category for early stage and new investigators. So if you connect with our team, so I can give you information on Randy King is the program officer, the program director, who manages this program. But what we've been able to do since between 2015 and 2019 is increase our early stage new investigators by 63%. We've more than doubled the number of early stage investigators who have R21s. We also have been, here's Randy. We've also been working with the National Institute of Aging. For those of you who are interested in neuroscience and Alzheimer's disease, there's been an enormous amount of investment in and seeking out new technologies and new approaches in Alzheimer's disease. So we've been able to award supplements to our investigators who write proposals. And this is a fairly informal process. Again, you want to get in touch with Randy. These supplements are due in March. We've been able to award roughly about 15, 16 of these supplements for the last couple of years. But we've been told by NIA, and I can't give you the numbers because I don't actually know what they are, but this will increase in 2019. So if you're working on a technology and you have NIBIB funding, or you have a colleague who has NIBIB funding, you should seriously consider submitting a supplement where you can pivot your technology to work on Alzheimer's disease. And this is getting this community inspired to work on Alzheimer's disease. I myself, like in the early 90s, got a $50,000 seed grant from the state of California to get people who'd never worked on breast cancer to start working on breast cancer. I knew nothing about breast cancer at the time. 30 years later, I had done a multi-center clinical trial in breast cancer with an entirely new technology that we built and designed from scratch. So you never know. Something like that could actually completely change the course of direction of your research. I want to advertise our debut challenge, the designs by undergraduate teams. So last year we had 52 applications. I don't think we had any from Purdue. I know we've had from Purdue in the past, So these are the undergraduate design team projects. We gave out $80,000 in 2019, and in 2020, we're up to $100,000 in prizes. Our first prize is the $20,000 Krosnick Prize. We've been able to get NIMHD, an institute that focuses on minority health and disparities, to sponsor a $15,000 prize for technologies in low-resource settings. So I'm hoping we'll see some great applications. These will be announced in the end of August, but awarded at the BMES meeting. So speaking of challenge, this is hot off the press. I think the notice of special interest is coming out and being published today. I hope it is. This is what they tell me. So this is a cooperation that we have with the Gates Foundation. This is a $1 million challenge. We just added NHLBI to that group. So us, Office of the Director, Allergy and Infectious Disease, NIDDK, the Fogarty International Center, and we just added NHLBI. So this is a technology accelerator challenge that is looking for the development of a platform technology or platform technologies that can solve at least one of these problems using non-invasive approaches. Malaria, sickle cell disease, anemia, so you need to work on one, but you need to solve two problems. So the rubric will be published soon, but I just wanted to get the announcement out there so people can start thinking about this. The idea is to develop a non-invasive platform device. Clearly, and there need to be two diseases in the vasculature that are being addressed. The Gates Foundation is our partner in this, and the applications will go through the NIH, and there will be an opportunity for the Gates Foundation to look at them in the next stage and possibly then provide additional investment and further development. But they're very serious and they're fantastic to work with, and we have a number of collaborations ongoing with them. Some of you may have seen the announcement from the NIH and the Gates Foundation together to cure sickle cell and HIV within the next 10 years. So this is gearing up. And this is a kind of a companion piece for that, because as you know, if you're going to cure a disease, you have to find and detect it, characterize it, and look at response to it. And that's a wonderful role that our community can play. We do a lot of meetings and bringing people together. Here is an ongoing multi-scale modeling meeting that Grace Peng, one of our program directors, has been organizing. So this meeting was held at the end of October last year. And in the fastest turnaround of a meeting to a publication, what they were able to do is get this paper published in a Nature journal on exactly what all of us are really interested in doing and expanding, using computational modeling combined with machine learning to improve our way to predict and understand biologic systems. So this paper, if you're interested in this general area, I really would suggest you take a look at this. It provides a lot of background and insight on the conceptual framework for combining machine learning with computational modeling, why and how artificial intelligence or machine learning approaches can enhance physics and mechanistic-based models, and where the limitations are, where the opportunities are. And it also does a really nice job of providing insight on some technical approaches. So this is a very fast-growing area. They have another conference that's coming up in March, and registration is open for this. This is an amazing community. There's such a diversity of activities and people and ambitions that are commensurate with the scale, from molecular and systems biology all the way to understanding how humans move and why they are directed to move in certain ways, and whether or not you can use that as a way of tracking and understanding whether they have underlying neurodegenerative diseases and so forth. So wonderful community. So let me try to sort of tie all of this together with the future challenges and opportunities. I've talked about how we have kind of engineered biosystems, whether they're molecules or cells that are on devices. So that sort of abiotic, biotic interface, organ simulators, tissue chips, molecules that are coming from a biologic process that are interfaced with silicon or fluidic. So we have an explosion of activity in this area. We also have an explosion of engineered devices that are literally put on and inside of biologic systems. So ASICs that you can implant, wearable optics and photonics devices and acoustic devices and electronic, bioelectronic devices that are designed to measure a variety of things and sense a variety of things. We also take humans, biologic systems that are complicated, and put them into giant physics engineered devices, like this illustration of MRI. And we've done really quite a lot of work in our community that not that many people are familiar with, but our community is quite familiar with the advances here, making computational models, using ODEs and PDEs and combining those with machine learning and representing how biosystems work. So where is all this going and how do you synthesize all of this? So we see this is fundamental to the emergence of the concept of digital twins. We know in engineering that digital twinning is ubiquitous in every new engineering project, whether it's a bridge or a plane, a building with many, thousands of sensors that are embedded. We have the capacity to be able to do this better for humans. What's been missing are the physics models of the human system, but those are now beginning to come together and they're becoming more and more validated, whether it's a microphysiologic system or an entire human. And we absolutely need this. There is no other way. We are asked by our colleagues in medicine to be able to understand and interpret all of this data, blood chemistry, images, genomics, patient's natural history. How do we integrate all that? It's impossible for a single clinician to take all of that and make it make sense. So this is where things are going. The development of digital twins, the ability to utilize this information to understand how people are responding to therapies, optimize therapies for individuals, predict the future for them, and virtual trials. So I have an image taken from a paper by Kyle Meyer's group at the FDA comparing digital breast tomosynthesis, a way of doing a kind of a digital mammography, to conventional digital mammography. So digital breast tomosynthesis is a 3D synthesized few views, not full tomographic view. And there is a big 10-year NCI trial called TMIST with 165,000 patients that's just looking at, is digital breast tomosynthesis better than digital mammography? So they're doing it the old-fashioned way, enrolling all those patients, taking all the images, comparing it to histopathology. So Kyle has published a 3,000 patient virtual trial where she's just her group has used radiation transport and radiation properties of breast tissue that are characterized and known to synthesize DBT, digital breast tomosynthesis, and digital mammography, and make those images and give them to human readers. Now, of course, you can use artificial intelligence and machine learning to read them as well. But in a short period of time, they enrolled 3,000 virtual patients on that trial. And you can imagine the cost difference between the 165,000 patient TMIST trial and the virtual trial is infinite. So these are where things are going in our community. We are the people that have to do this. And there are huge opportunities to really change outcome for patients by being able to bring those tools together. The NIH, if you're skeptical, has already said this integrative approach is the future of health. There is this program, if you haven't heard of it, called All of Us, which is another common fund program. And it's designed to enroll, actually this is not common fund, this is directly appropriated, I believe. So this is a huge program that's enrolling at least 1 million subjects designed to really understand the trajectory of individuals with their health. And it's using kind of that standard approach of snapshots, static images taken over a long period of time. So if you want to make the movie of life and disease processes and responses to therapies, you have to have 1 million or more people because you're taking static snapshots of many, many individuals and then you have to put them together. But that's a blurry movie, I would argue. And what our community can do, our opportunity, is to make that movie in individuals. We don't need 1 million people, but we need to develop the technologies to get the dynamics in just a few individuals so that we can see those processes better. So let me try to summarize. We've had lots of growth in bioengineering. We're now 13% of the entire NIH budget. And our community is really well integrated into many other communities. People are looking to us, and it's our responsibility, if we do so want to see a future of health, like all of us is hoping and envisioning, we really have to get into the mix. There is huge potential here where the technologies we're developing are fundamentally cost-saving technologies. Imagine virtual trials and digital twins, microphysiologic systems to represent us so that we can optimize therapies for individuals. And we can unlock some of the mysteries of the dynamic biologic processes and do a better job of predicting outcomes based on dynamics, not having to look at static processes over very long periods of time. The future needs a new workforce, and hopefully that's a lot of you, and you'll continue to pass that on and those training techniques. And the interesting implication here is with these kinds of tools, we can shift to prevention and optimization. Really think about optimizing health span, preventing disease in the first place. So health span and lifespan are sort of ideally matched. These ultimately, we think, can reduce costs and further drive technologic innovation. So engineering is essential. That's the picture. And I have a little video. It's sort of, I'd like to, my advertisement for some of the things that are going on in our community. And I'm hoping that in the future we'll add some Purdue technologies to this, or Indiana technologies. Okay, there you have it.
Oh, it's fantastic. A question over there, would you use one of the handheld microphones, because we have overflow rooms, so they can hear your question in the other rooms.
Hey, Umberto Tacchinardi, IU School of Medicine and Regenstrief Institute. Thank you so much for this impressive and exciting presentation in terms of our future. My question is, I'm in the informatics world, so I was very happy in one of your first slides when the Congress findings pointed informatics as one of the areas that are required to be supported in this new era. I didn't see, and you pointed at some interesting challenges like development of platforms that can support data integration in a very sophisticated way for this new world of science and development. My question to you is, how do you see how the NIBIB is seeing the evolution of algorithms and completely software-based devices connected to EHR systems as part of their mission? Because right now, it's my impression that the NIH is still to define a more clear path for areas that are not specific bioengineering and are not data science, bioinformatics, and
Yes.
I would appreciate your comment on that.
Yeah, this is a really good question. So we have a strategic plan in data science and a scientific data council. We have a new initiative to move data all across the NIH and in the extramural community to the cloud. And there is now, this initiative is called STRIDES, to make it findable, accessible, interoperable. So there is now 30 petabytes of data. There'll be a single sign-on. And we have released a few notices of special interest to stimulate the development of new tools that would also potentially be commercializable. So one is an SBIR, STTR announcement. And I would say from the point of view of innovation and new approaches, NIBIB is the place to go. So if you're introducing novel ways to analyze, extract information, predict future outcomes, synthesize and integrate multiple information together, that's kind of an NIBIB domain. In fact, a lot of encouragement in the use of machine learning and artificial intelligence there. We actually spend almost 12% of our budget, more than any other institute, on AI and ML-based approaches. But fundamentally, many of the things that we do are grounded in physics and mechanistic-based models, and then the machine learning is an enhancement of those. So I would say other institutes are also supporting many of these things, but not so much from the novel tools development. So NIBIB would be the place to go to for the novel tools development. I can connect you with the right program directors. So Behrouz Shabastari is very, very interested and supports kind of a health informatics type of portfolio. But there isn't that much innovation. You know, a lot of it is just the discovery, as I've looked across the NIH, that now there is commercially available software that does things that, you know, were not possible to do 5-6 years ago. So you see a lot of people integrating that in that, and the innovation component from the computational algorithm development side is not that strong. So I would definitely encourage and recommend NIBIB as a partner for that. Yes.
I'll get Purdue BME. I have one question about NIBIB, also NIH in general. The current review process, I think the future depends on young scientists, not the old one like me. But young scientists need to have research funding to do their research. But as I travel the world, American research funding, no matter how many $1 billion you spend, young faculties do not have enough funding. But Chinese counterpart, at least 10 times more money than America. To compete with all of the world for the future, we have to invest in young scientists. But I don't see how NIH is spending money for young scientists. Most of the money goes to, NIH director mentioned last year, most of the senior researchers got 40% of total money. That's unreasonable, and also it's not sustainable. So what is your plan to support young scientists for the future? Because they are the future.
Yes, so that's a great question, and that was one of the reasons why we started that Trailblazer program. So that's had a very significant impact on our new investigators and early stage investigators. And across the NIH, this is a topic that's very, very frequently discussed and analyzed. I don't know what the latest numbers are, but I know that we're improving in funding young investigators and early stage investigators. There will be a new initiative that comes out that's, I don't know if it's completely finalized yet, but there will be an entirely new initiative that looks a lot like that Trailblazer Award that will go all across the NIH. It's going to be an entirely new type of R01 mechanism that's specifically for new investigators. So I can just say in discussions I've been in with all the institute directors and the leadership at the NIH, there is a lot of emphasis on developing new programs and trying to correct that course, because you're correct. It is a point of concern and has been for some time. And then, you know, we also are looking at middle stage investigators, because that becomes another, you know, issue. We've been fortunate to have, over the last few years, some modest budget increases across the NIH. And I think we suffered when there was the time 5-6 years ago when budgets were flat, in fact even went down. We were not able to actually have budgets even coming from Congress. So because of a little bit of the politics behind the scenes, that not everyone understood the ripple effects in the broader community. But over the past few years, this has now improved, and we're able to introduce some new mechanisms. So if we have young investigators who are having difficulty getting their funding for their ideas, it's important that their voices be heard. So their faculty mentors, they themselves should connect directly with program directors at institutes where they think that they're best matched. We see there's variation across the NIH. For example, the National Cancer Institute, if you come there with an R01 grant, if you don't score better than 8th percentile, you may not get funded. Our pay line last year at NIBIB was 19th percentile. And if you're a young investigator, we add 5 extra points. So if you're 24, 25, 26th percentile as a young investigator within the NIBIB, then you will make it in the pay line. So NIGMS is also has a much more generous pay line for R01 investigators, and for young investigators, it's moving in the mid to, I don't know exactly what the numbers are, but I think they're even higher than ours. So the pay lines have changed. And the perception of young people, you know, when I was on panels and workshops 5 years ago, there's a lot of despair, but not just from young people, from everyone, because of the things that were going on with the budgets. But I think things have changed over the last 5 years, and I think statistically you can watch those increased appropriations and their impact, and how they've changed the pay lines. So right now, things are, I'm cautiously optimistic. In fact, NCI just got an appropriation from Congress to address the difficulty of their pay line, to try to expand it. The reason why their pay line is so low is because they're heavily invested in large centers, their cancer center programs. And so that occupies a big chunk of their budget, so they don't have as much flexibility for individual investigator grants. But through partnerships, we partner with the NCI to promote and develop bioengineering technologies. So some of those are also good areas to look. It's a massive, massive organization, $40 billion. And I feel like it's really kind of like a giant starship. There's so much going on. And it's important to engage program directors, engage people inside of that organization to help understand and navigate through it. Go to workshops. Back here.
With the increasing and important commercialization activities involving the academic world, do we need to worry about conflicts of interest in the review process?
So the question is really, I think, about when you submit a grant proposal and whether or not that implicitly there is I don't know what the sort of the built-in expectation of that is with respect to disclosure. And I'm not an expert on sort of how Center for Scientific Review manages those potential conflicts. For sure, there is a conflict evaluation stage in the review process, and people are excluded for conflicts when people are being recruited to do review. There's a weeding out at that stage, and then in the discussions in the panels. But I can't really address that probably as well as you would like, since I'm not fully aware of all of the steps that are taken to eliminate COI in that process. But it has, from my understanding, it's worked reasonably well. But the numbers are staggering. The number of applications that come in, I think, on the order of 70,000 per year. Just the massive throughput in this process.
Move on to the speaker. Thank you so much. Great talk, Tommy Source with Purdue University. The question is, how do we get more involved with the RFI process? How do we engage more with you and your institute to be part of the conversation at the beginning, so like that we can also prepare ourselves for when funding opportunities might come up? What's the best strategy that you would suggest?
So I think just exactly what you're doing, much of that happens. So first we don't have I'm still learning the nomenclature. There are not that many set-aside programs. I think much of what the NIH has moved towards are what are called notices of special interest. What you see that's quite visible in terms of set-asides are programs that are created from what's called the Common Fund, which is a fund that the Office of the Director manages. And those initiatives that come out of the Common Fund are launched much like you collaborate with your colleagues. Those are launched through the collaboration of institute directors and program directors within the NIH. And the requirements are the idea has to be something that many, many institutes are really excited about. So for example, I co-led data science and innovation in Africa, and that really hit a nerve. There are 19 institutes that have signed on to that. They all think it's a really fantastic idea. You should check it out, the description of it, on the Common Fund website. The Spark Initiative, another Common Fund program. So that involved collaboration amongst several institutes in order to pitch it and sell it to the overall director of the NIH. So other things that you'll see are what are called notices of special interest, and they're published in the guide on a regular basis, if not weekly, maybe even at higher frequency. And those are really just kind of statements of interest from institutes and people across the NIH who are saying, commercialization in informatics for the development of algorithms that will help us reconcile FHIR standards. FHIR is Fast Healthcare Interoperability Resource. And you can give a lecture on that. But it's increasingly important that if any of that vision that I talked about for the integration of multiple disparate information content that comes from a variety of sources, if electronic health records will be part of it, it's important to have standardization. And FHIR, which is an entirely volunteer organization, is establishing and setting those standards. So we recognized that, and we published a notice of special interest that said FHIR is important. And we have a series of them on FHIR. And we've also, from our institute, published one that said we would like to stimulate innovation in the use of FHIR standards for API development, for example, and then have SBIRs and STTRs that people could apply for, because we see that's probably, that drive is the only way that there really is going to be integration and true access to that information content. So that's kind of the process. I've just integrated a lot of different sources of information, but there is a kind of ongoing conversation that you can visualize through the guide notices. And then you can respond. Now there will be RFIs, requests for information. So for example, when we get our first draft of the strategic plan, we'll publish an RFI. And hopefully I'm correct in this because I'm still learning the language. I think that that's when we ask for comment from you. So that our community will then, it's kind of like a Dear Colleague letter from the NSF, where you get to comment and well, maybe it's not the same as a Dear Colleague letter, but you get the idea. We'll send stuff out and then invite the community to comment. So those things are ongoing all the time. I hope that answered to some extent. It is you can see it's confusing.
Theresa from ECE. I'm assistant professor. And thank you, first of all, thank you for the fantastic talk and showing us the opportunities as well as the history of how NIBIB emerged. And as you write, technology is going to empower some of the future innovations. So one of the technology we see electronics, being able to make it energy autonomous and reduce the form factor so that it gets to a almost invisible form factor and be part of a lot of the transducers that are being built, for example, the acoustic array. But if you look at all of these transducer papers, basically the electronics is still off chip and they don't show that in the paper. It's basically tethered and outside. So most, okay, so most of my money comes from. But if I try to come to NIH, I have seen that I would have to cater it towards our application. Whereas the actual science that is going on is very generic towards all or many applications. So is there first of all directorate which funds future of electronics for biomedical applications? Or is there a future effort to combine with NSF and DARPA with NIH? To think of something like that in the scale of what imaging did for last 30 years for miniaturized electronics for next 30 years.
Yes. Okay, this is a really, really good and important question. I hope everybody heard it, but I'll kind of repeat it. Do we have an NSF or DARPA analog within the NIH that's helping facilitate the development and addressing the challenges, the true challenges of miniaturization with, particularly in my universe, we move towards the development of ASICs. And so, imagine, and there's a lot of ASIC activity, they can be used for a variety of different things. I can put a biologic molecule, I can put an aptamer on an ASIC and have a sensor for a single molecule. And then I could implant that in an organ. So, right? So the short answer is NIBIB. We have that in our portfolio. We are non-hypothesis based, so disease agnostic. The only thing you have to do is say, this could work for a variety of different things. For example, it could help in diabetes, but it's metabolic, and it could be useful for other metabolic diseases. So that's all you have to do, and we do work with NSF. We fund grants that go into the cyber-physical system, so we've signed on to that. We communicate regularly with DARPA, particularly through the BRAIN Initiative. And you have to differentiate. Why would it go to the NIH, and why would you need those resources as opposed to whatever from the NSF or from DARPA? But I think there are many, and we can, if you want to, we can talk about obvious directions there. We have several program directors who have exactly this expertise. So you want to connect with them and talk to them on how you get a NIH sort of, what you really have to do is you have to get through the study sections at the NIH. Unless it's a special study section that's created for a special initiative that, for example, NIBIB puts together. So that's kind of the hurdle. So it's not necessarily always the institute. Now, how do you do that? Again, the roads go back to the program director, because the program directors understand the study sections. They understand what's been coming through them, and they understand the culture of the study section. So that, that's an important process. So I'm happy we can talk more, and I can connect you with more people. But I want to absolutely encourage that, because look, we're like at 10 nanometers or so right now in integrated circuit in transistor size, and we're approaching the limit. But think of all that capital investment to get there. And I don't need 10 nanometer structures to be able to have the next fantastic wearable sensor that's multi-parametric that will report on everything I need in order to predict the future of a patient who has heart disease or diabetes. I absolutely don't. 100 nanometer would be fine. So we have all of this capacity to repurpose that amazing effort that's gone into consumer electronic devices. And then that's the abiotic part. All right, imagine coupling that now with true biologic systems, whether we take the biologic system and put it on the chip, or we put the chip in the biologic system. Yeah, I'm completely with you on that. That's enormous opportunity for growth. We won't have the future of health without it.
So just to add, so do you see NIBIB supporting that growth, or a parallel similar organization being created within NIH?
We are already supporting that.
Okay.
We are absolutely already supporting it. But again, it sort of all comes back to, in any of these, you have to understand the process. How did the ones that we are supporting, how did they get to that point? So there's both the technology itself, and then there's understanding how the technology got into that system and got to be supported. So that's what we have to help educate our community on.
Thank you.
One more question.
Pavlos Vlachos, Mechanical Engineering. Again, thank you as well. It looks like NSF is testing a new model with the Convergence Accelerator, if you're familiar, where it kind of blends an entrepreneurial type of high-risk spirit with a mission-driven, expedited DARPA, get things done approach. I'm just wondering if NIH is considering an experiment like that, and if so, maybe you can break the news and we can get involved early in the process?
Yes, so this is a great question, because as all of you know, especially if you've had an R01 or familiar with the R01 mechanisms at the NIH, the R01 is not really ideally matched for the design, test, build, deploy cycle that we have in our device world and our technology community. We need other mechanisms. So we are for sure exploring that within NIBIB to try to better match the way that our community actually works and to meet the needs of our community. The R01 is a mechanism that's available, but there are others. So the R21 may be an alternate path, and that's why we created the Trailblazer Award. But there are others. So there's what's called the Other Transactions mechanism. That's a DARPA mechanism. And the SPARC program is funded entirely through the Other Transactions. There is a limit by federal law on the amount of money that can be distributed by Other Transactions. So we're in close contact with the NIH director on that, but that's another really interesting approach. And then there are challenge grants, like we've created, and the debut awards for undergrad students is a kind of prototype test of that. That's been very successful. Now we're sort of moving to this next challenge grant in cooperation with the Gates Foundation, which will have up to $1 million in prizes. And it's speccing broad design characteristics. And anybody can come in with a concept or an idea, and that potentially changes how devices and technologies would be funded. So we're absolutely exploring that and cognizant of the fact that our system is a little bit rigid. We're also partnering with NSF, and in particular the Industry and Innovation Division, and looking at ways that we can do a better job partnering with them, possibly with the IUCRC approach, and also at the other end of things with the ERC approach. So absolutely, we've got to improve the development side as well as reduce barriers towards commercialization, dissemination of devices, kind of on the other end, which involves really addressing some of the regulatory issues as well. So yes, we have a broad mandate to rethink exactly how we're doing stuff. And I'm very interested in hearing your ideas on what works and what doesn't work, and how we can improve.
Okay. Thanks. Let's thank Dr. Tromberg again.